All right, we're going to get started on the fat lecture. I'm not going to go through the eicosanoids, the types of fats, because those are fairly straightforward, but instead going to start here with digestion. So not shown is lingual lipase. This is in the mouth. And recall that if it is a lipase, it's breaking down fats. Lipases specifically are breaking down fats into triglycerides usually. Triglycerides, some free fatty acids. So if we look over here, we have our emulsion droplet or fat droplet. When it's in the stomach, the gastric lipase is released and we continue our fat digestion. So if we look here, we have fatty acids that some of them are free. Gastric lipase is on the surface. It is trying to break down this fat droplet, but also recall that if, especially things that are ball-shaped, the larger equals decrease surface area to volume ratio. So this, even though it's a large droplet, has less surface area to its volume. So it's hard for these lipases to get in here in the center. But between these gastric lipases and motility, the stomach breaks down large fat droplets into smaller ones. So now we are in the intestines, specifically in the duodenum, and the pancreas, not pictured but now drawn, releases pancreatic lipase. We also get colipase, which is going to help activate, and we have bile acids. And so bile acids here, they're called bile salts, but these are going to further break down this slightly smaller fat droplet into fatty acids, into 2-monoacylglycerol. It's going to diffuse into micelles in the, or this fat droplet is going to become a micelle, which can diffuse in the duodenum and jejunum. So here we have a micelle. And micelles are made up of fatty acids and some triglycerides, less triglycerides and more of these diacylglycerols and monoacylglycerols. These micelles are now small enough that they can actually just diffuse into the enterocyte. And so that's what we see here. We also see absorption of these monoacylglycerols, free fatty acids. These are all small enough to just diffuse into the cell. And we're able to do this because our cells have phospholipid membranes. So that's all of the what I would call gross digestion. That is kind of a large fat droplet getting broken down into a manageable micelle, which is then taken into the mucosal cell that we see here. So here we see that we have one of our monoacylglycerols, and we have 2 free fatty acids. Combined, this would be a triglyceride. one triglyceride. It got broken down into a monolysyl glycerol. It got broken down into 2 free fatty acids. And the R here indicates that we don't know exactly what type. The R group is just how many carbon-hydrogen moieties are comprising the tail. So just remember that this R group, it could be something like, and then have H's, depending on its saturation. If it's fully saturated, then there's H's all along here. This would be a fairly short one, but this is just an illustration. All right, so our monolysyl glycerol, our free fatty acids, are going to diffuse into the brush border membrane. And just like carbohydrates, we need to tag these with something. Otherwise, they are just going to diffuse back out of the cell. So in carbohydrates, we tag our sugars with a phosphate group. For fats, we tag them with an acetyl-CoA, and we do that through CoA synthase. Or we tag them with a CoA from an acetyl-CoA, excuse me. If you look, this requires energy. So here's the CoA group. We use an ATP, and we put that CoA onto our free fatty acids on the carboxyl end. Then we can take these free fatty acids, and within the mucosal cell, we actually repackage them into a triglyceride. So here we see, here is the glycerol backbone. This is our first fatty acid tail, second fatty acid tail, third fatty acid tail. From the mucosal cell, we now take this triglyceride, it goes into the lymph. Then it is packaged into a chylomicron, and chylomicron is a lipoprotein. And lipoproteins can be transported through the blood. We're unable to transport a lot of triglycerides in the blood without a lipoprotein shell, for the same reason that salad dressings tend to separate. Our blood is predominantly water, and these are obviously fats, so if we didn't put them in a lipoprotein casing, they would separate out of the blood. So then, in the circulation, in the bloodstream, these chylomicrons will, I like to think of them as beach balls. They will bump into lipoprotein lipase, and lipoprotein lipase is going to take triglycerides out of the chylomicron. And this is very fast. Chylomicrons are cleared within 5 minutes. So let's remind ourselves what a chylomicron looks like. These are all lipoproteins. And they get this name because they are made up of a phospholipid membrane that has proteins embedded in it. And these proteins actually tell the body what type of lipoprotein this is. So as you can see, chylomicrons have these lipoproteins that have various names, B48, AI, A1, A4, A5, whereas a very low density lipoprotein has the lipoproteins B100, C, and E. You don't need to know all of the different lipoproteins, but what you do need to know is that a chylomicron has different lipoproteins than a VLDL, but the LDL and VLDL have similar lipoproteins. They both have a B100, and then the HDL has different lipoproteins from the LDL and VLDL and the chylomicron. So we can kind of think that the proteins are what makes these lipoproteins different from each other. Okay, let's now look at the inside. These yellow dots, these are triglycerides. So as you can tell, a chylomicron is made up of a lot of triglycerides, high triglycerides. A low density lipoprotein though, very low density lipoprotein, has fewer triglycerides. So because it has fewer triglycerides, the concentration of cholesterol esters is higher. I haven't actually counted these before. Let's see. 1, 2, 3, 4, 5, 6, 7. So there's 7 cholesterol esters here. Let's see how many are in the VLDL. 1, 2, 3, 4, 5, 6, 7. So these aren't exactly equal. This VLDL has slightly more LDLs, but basically, as the chylomicron donates its triglycerides, the overall proportion of cholesterol esters increases. And you'll actually see that we have cholesterols themselves in their final cholesterol form embedded in the phospholipid layer. And similar to in our cell membranes, cholesterol in the cell membrane will change the fluidity of this membrane. All right, if we look at an LDL compared to a VLDL, the LDL has almost, well, in this one, it has no triglycerides. The LDL particle has only cholesterol esters, all blue. So that's why the LDL is what we measure when we measure cholesterol. The HDL also only has cholesterol in it, but as you can see, it's much smaller, it's more dense, and it actually is picking up cholesterol esters where the LDL is donating cholesterol esters. Let me erase some of these marks before we go on to the next diagram. Okay, so this diagram is showing a lot. So we're going to walk through it. And we're going to start in the intestines. So this assumes that we have eaten something high in fat. And we have our lipid droplet, all of that good stuff happens. If you don't remember the digestion, go back a couple of slides. So now we are in the intestines, and we put our triglycerides into a chylomicron. Again, the details are on previous slides. And these chylomicrons are in circulation, so these are in the blood. And chylomicrons can go a couple of different places. So the chylomicrons can go from the intestines, and they can go to, they can go to a peripheral tissue. When we have peripheral tissues, think fat tissue, think muscle tissue. And these tissues have lipoprotein lipase. Again, lipoprotein lipase is clipping triglycerides and free fatty acids out of the chylomicron. So it clips these out. Here we have a free fatty acid. It can be used in the peripheral tissue for energy. But if it's in a fat cell, it's probably just going to get stored. And we're actually going to rebuild our triglycerides for storage. So something you'll probably notice about fat metabolism is we go back and forth between disassembling and reassembling fats over and over. And this is primarily because fats are difficult for us to transport because they're hydrophobic. So if our chylomicron doesn't donate everything it has to the peripheral tissues, it gets sent to the liver. So let's say this was 1, this was 2, here's 3. Ooh, that's a bad 3. All right, so we have what's called a chylomicron remnant, meaning it has donated some, but not all, of its triglycerides. This chylomicron is then going to be taken up through the liver, and it is going to get repackaged as a very low density lipoprotein. So we had excess triglycerides and cholesterol esters. The chylomicron gets repackaged into a VLDL. And it's different from a chylomicron because it has different proteins. In fact, it gets tagged with an ApoB100. Here we see the initial chylomicron got tagged with a B48. So, in the liver will release that very low density lipoprotein. And again, this very low density lipoprotein is going to hit LPL on peripheral tissues. LPL is going to do the same thing. It's going to bring in fatty acids and triglycerides, and these are either going to be used for energy, but more likely if it's in fat, they're going to be stored. So that VLDL will then donate the triglycerides until it becomes an LDL, or an intermediate density lipoprotein that we don't have a picture of, but you can imagine its density is between these two. So this VLDL remnant will get taken up by the LDL receptor. The LDL receptor is able to tell the difference between the chylomicron remnant and the VLDL remnant, again, because of these proteins. And you don't need to know the proteins, you just need to know that the reason the body is able to tell the difference is because a VLDL has a different lipoprotein than a chylomicron. So this LDL is taken up, and that is all good. It can be broken down into triglycerides. Those can even be repackaged again. The problem occurs, and we see that in the red mark here, when we have too many VLDL remnants, and now we're in the overflow pathway. We have low density lipoproteins, excess low density lipoproteins in circulation. So we also have low density lipoprotein receptors on our peripheral tissues because we need a little bit of cholesterol. Think about why we need cholesterol. We need it for bile acids. We need it to make lipid hormones. We need it for cell membrane stability. But this is when we have too many. These LDLs are taken up in the peripheral tissues, and their cholesterol then gets either put to use, or ideally, it gets picked up by a scavenger HDL. But again, when we have way, way too many LDLs, they're going to start to build up in the arteries. When they build up in the arteries, we start to get plaques. And the body recognizes a plaque as a problem, and it will send an immune response. Unfortunately, this immune response generally just increases inflammation, and that's going to make the artery close, so increase closing. And this is especially problematic because HDLs can only pick up so much extra cholesterol. So if we're eating way too much fat, we're going to create excess LDLs. We also will end up with excess LDLs if we're eating too many carbohydrates. So just a brief overview that we do have different types of fat cells. The ones we're probably the most familiar with are the white adipocytes. And white adipocytes have a big fat droplet. And for a while, these were almost considered completely inert. They're not completely inert, but they have a very low metabolic rate. They don't need to contract. They aren't functional in the way, they're not an organ tissue that is metabolically active, like the liver, although they are a tissue that is active, but they're less active. And so we only have a couple of mitochondria and a nucleus. The white adipocyte is primarily this fat droplet. And remember that if the fat droplet increases, this is hypertrophy. And if we have more fat cells, this is hyperplasia. Okay, let's erase this really quick. So the other one you might be familiar with is the brown adipocyte. So notice the fat droplets are small. And there's a lot of mitochondria. The mitochondria is actually what's giving the brown adipocyte its color. And if we think about muscle cells, think about the difference between type 1 and type 2 and type 3 fibers in muscle. They have different colors, and again, part of that color is due to how many mitochondria they have. Along with the more mitochondria, the more blood flow you need. So brown adipocytes have small lipid drops because this increases available surface area for the mitochondria. Also remember that in brown adipocytes, they are doing non-shivering thermogenesis. And they're doing this through uncoupling proteins. They're uncoupling OXPHOS. That means they are decreasing the proton concentration gradient, and they're generating heat. This process is what makes brown adipocytes more metabolically active. It's the activity of these mitochondria. And it's not in the same way as muscle contraction, where the mitochondria are using energy to contract the tissue. The mitochondria are actually uncoupling from oxidative phosphorylation and generating heat. mitochondria. So additionally, we have an intermediate between white and brown adipose tissue, and it is kind of conveniently named beige adipocyte or tan adipocyte. And this is, as I mentioned, an intermediate between white and brown adipocytes. You can see they have some kind of larger fat dropules. They have some small fat droplets. They have more mitochondria than a white adipocyte, less mitochondria than a brown adipocyte. And so, similarly, they are kind of in between a white and brown adipocyte in terms of metabolic activity. The interesting thing about beige adipocytes is that beige adipocytes can be induced through cold exposure. But this takes a long time. Much longer than most people want to be cold for, and even longer than those cryo-sculpting companies would have you believe. So last but certainly not least, we have the pink adipocyte. And it's called this because the pink adipocyte produces a milk droplet. So we find these in breast tissue and mammary glands. We see that they have some intermediate-sized fat droplets, but much, much smaller than a white adipocyte. And in addition to mitochondria and the nucleus, we also see that there's a rough endoplasmic reticulum and a Golgi apparatus. And if you remember from your Life 1010, the Golgi apparatus and rough endoplasmic reticulum are packaging organelles. So what they're packaging are milk droplets. So these mitochondria, the Golgi apparatus, the rough endoplasmic reticulum are working to put fats into milk droplets so that those milk droplets can be secreted and consumed by what I assume is a hungry baby. So one note I want to make here, this is a misprint. There shouldn't be a lipase here. This is a diacylglycerol, just like a triacylglycerol or a monoacylglycerol. There is a monoacylglycerol lipase, and it's here, monoglyceride lipase. So, apologies for that. This is not, there's no diacylglycerol lipase. It's called hormone-sensitive lipase. Okay, so this is inside a cell. We have a large cytoplasmic lipid droplet. That's what the C stands for. And from this lipid droplet, we can do a couple of things. Number one, we can use the lipids from the lipid droplet and we can send them to the mitochondria for energy. And so we see this is an autophagosome. It is taking some of the lipid. It's then putting it through a lysosome and clipping out free fatty acids. These free fatty acids, again, we always have to put a CoA on them. Otherwise, these guys could just leave the cell. But here, we are putting them into beta oxidation. And from beta oxidation, we can create our acetyl-CoAs. And acetyl-CoA can go into the Krebs cycle and to oxphos to create ATP. So that's path one. One, we can use lipids for energy. Two, we can create a new lipoprotein. and this is in the endoplasmic reticulum. To do this, we need to take a triglyceride from the cytoplasmic lipid droplet, and we need to clip it into glycerol and free fatty acids. So to do this, we're going to use a series of 3 lipases. The first lipase is adipose triglyceride lipase, ATGL. ATGL clips off 1 free fatty acid. That takes a triacylglycerol and it makes it a diacylglycerol because now we have 2. Next, we have hormone-sensitive lipase. Hormone-sensitive lipase, right here, is again going to clip off 1 triglyceride, and we're going to get a monoacylglycerol. From a monoacylglycerol, we then can have, which it isn't shown here, but it is 3 monoglycerol lipase. And unsurprisingly, monoglycerol lipase is going to clip off 1 fatty acid. And now we're left with our glycerol, and we have 3 fatty acids. Of course, if we don't do this, they're going to diffuse out of the cell, so we have to we have to put a CoA group onto them from acetyl-CoA. So CoA, CoA, CoA. And now we can take this fatty acid and we repackage it. We get a glycerol 3-phosphate. We could get that from this glycerol. We could also get this from carbohydrate metabolism. And then we do this process backwards. We put a triglyceride, or we put a free fatty acid, onto our glycerol backbone. We then put on a second free fatty acid, and now we have a diacylglycerol. We put on now we clip off the phosphate group, actually, that's this step, but after that, we get a diacylglycerol. And now we can do the final step, put on our third free fatty acid, and we have a triacylglycerol again. That's what we started with. Now we can put that triacylglycerol into a lipoprotein and send that to circulation. So that's our second possibility. Third, so again, we have our triglyceride. We use adipose triglyceride lipase to clip off 1 fatty acid. We have a diacylglycerol. We use hormone-sensitive lipase to clip off another fatty acid. And then we use our monoglycerol lipase, number 3, and that clips it into the final free fatty acid and glycerol. Again, we put on a CoA group, and this is also a signaling molecule. So this can go into the nucleus, and it can activate PPAR alpha, which is one of those genes that's related to lipid metabolism. And if it's PPAR alpha, it can activate genes in lipid catabolism. If it's PPAR gamma, it can activate genes in lipid anabolism. So this would be breaking down lipids. This would be building lipids. So that's option 3. finally, also with option 3, because these are signaling molecules, we can also create eicosanoids. Eicosanoids, if you recall, from the beginning of this lecture, these can be thromboxanes, prostaglandins, and leukotrienes. So, something to keep in mind in this process is that adipose triglyceride lipase is the control mechanism of this lipolysis. So right here, put stars by it. This enzyme is regulated by insulin. If we have low insulin, we have high adipose triglyceride lipase expression. Think about what insulin is signaling. Insulin is telling us if we have nutrients or not. If we have nutrients, insulin is going to be high, and it's going to signal for storage. So insulin equals storage. If insulin is low, that means we don't have anything to store, and it means we need to use our lipid droplets for energy. So that previous diagram only looked at if we had lipid droplets only looked at lipids from the lipid droplet. But we also have exogenous sources of lipids in the blood. We can get lipids from chylomicrons, and recall this is through ApoB8 or ApoB48. We can get cholesterols through our LDLs and ApoB100. We can get some triglycerides from very low density lipoproteins, and so as this is donating triglycerides, it's becoming a low density lipoprotein. And albumin also will carry free fatty acids, not triglycerides, just free fatty acids. So if we have either a chylomicron or very low density lipoprotein, LPL is going to clip off a free fatty acid, and here LPL is going to clip off a free fatty acid. Whoops. These free fatty acids, again, going to get tagged with a CoA group so that they can't diffuse back out of the cell. So we have two things. Depending on how much we have consumed, we can either store or we can use. If we have enough exogenous sources of glucose, we don't need to use this. We need to store it. So if we're going to store it, you'll notice it goes back to the endoplasmic reticulum, and it goes through the exact same process. We're going to attach it to G3P. So we're going to take that free fatty acid, we're going to put it on to G3P. So now we have a phosphorylated, essentially monoglycerol. We're going to take a second fatty acid, we're going to put that on, still phosphorylated, so then we're going to remove the phosphate group and we have a diacylglycerol. And then we're going to take our final third fatty acid and build a triacylglycerol. And then we're going to store that triacylglycerol in our cytoplasmic lipid droplet. That's the store pathway. The store pathway is going to be higher if insulin is high. So, if we have low glucose, we're going to need to use some fats for energy. Those will probably come from the lipid droplet. Additionally, if we think about, let me erase this really quick. If we have a high concentration of glucose, that means we're preferentially going to use glucose. So it's going to go through glycolysis. It can, then it'll become pyruvate. We can also get pyruvate from amino acids. This is kind of a hint to the next lecture. That pyruvate can go through the TCA cycle and become energy. But if we have enough energy, it's going to turn off this cycle. And instead of producing energy, this pyruvate is going to go towards de novo fatty acid synthesis or de novo cholesterol synthesis. So this is when I mentioned an excess of glucose will increase cholesterol. This is how that happens. And this cholesterol ideally would get picked up by a low-density lipoprotein, but it could get picked up or used, or we can put it into other lipoproteins. So, in summary, if we have a lot of glucose, we're going to store any fats that we're consuming. Insulin will be high. If we have so much glucose that we can't use all of it in the TCA cycle or in oxphos, we're going to put it into de novo fatty acid synthesis. In de novo fatty acid synthesis, we're then going to probably go store these. We also can make cholesterol. And that cholesterol can then be used, or if we have too much, that's when we start having problems. It's going to start building up in our arteries. So we've spent a lot of time looking at low carb or low fat. In fact, that's what your abstracts were about. We've just had several lectures on fat and carbohydrates. So which diet is better? Well, a researcher at NIH named Kevin Hall looked at this, and this is his paper, and he found calorie for calorie dietary fat restriction resulted in more body fat loss than carbohydrate restriction. So dietary fat restriction is important, and it will lead to more body fat loss than carbohydrate restriction. And how he tested this, he gave 19 adults either a high fat diet or a low fat diet, restricted calories by 30%, or he restricted carb intake. So decrease of calories from carbohydrates. So decrease fat, decrease carbohydrate. And he found that cutting carbohydrates increased fat oxidation, but it had no difference on carbohydrate oxidation. But cutting fat resulted in more body fat loss. So let's unpack that. They measured this through metabolic balance. And they did this by calculating the daily fat balance as the difference between fat intake and fat oxidation. So that's fat oxidation or actually it's fat intake, minus fat oxidation. And so if we look at their RQ results, recall that RQ is equal to carbon dioxide divided by oxygen consumed. So this is consumed, and this is expelled. So people on a reduced carbohydrate diet decreased their RQ. If we have a lower RQ, low, that means that CO2 is smaller than O2. With the reduced fat model, we see no difference. So this was a run-in time, and we see this decrease in RQ that reflects eating more fat. And eating more fat requires more oxygen. But in the reduced fat, this is a higher carbohydrate. We don't see a difference in the respiratory quotient. Makes sense because we are eating more carbohydrates in that. So fat oxidation, like I mentioned, is measured using RQ. And RQ, or is measured using indirect calorimetry. Indirect calorimetry is measuring the volume of oxygen consumed, the volume of carbon dioxide expelled, and they also had urinary nitrogen. And this is to account for protein intake. And they also looked at net carbohydrate oxidation. Again, has the same inputs, oxygen, CO2, and nitrogen. And they looked at protein oxidation, and that is just based on nitrogen. So they then used this fat oxidation, and they looked at how much fat was consumed. And fat balance is then fat intake minus fat oxidation minus de novo lipogenesis. So this is making lipids, mostly from carbohydrates, CHOs. Carb balance is carb intake minus carb oxidation plus gluconeogenesis, because this is new CHOs, and minus de novo lipogenesis. We're subtracting de novo lipogenesis because these carbs are now being used up here in fat balance. Finally, we have protein intake minus protein oxidation from here minus gluconeogenesis. And this is because gluconeogenesis uses protein to make carbohydrates. So they did a lot of different measurements. And let's look at these one by one. If we look at fat oxidation, the reduced carbohydrate data and model oxidized more fat. So let's say reduced carbohydrate, oxidize more fat. Let's look at carbohydrate oxidation. The reduced carbohydrate diet oxidized, less carbohydrates. And between the two diets, really no difference between protein intake, because protein intake was held constant. As part of the diet, but in the reduced carbohydrate diet, there is slightly more oxidation of proteins. And this is important, even though it's not significant. It is higher. So let's look at fat balance. So the change in fat balance is higher in the reduced carbohydrate. Carbohydrate balance is lower in the reduced carbohydrate diet. Makes sense, there's less carbohydrates to oxidize. And protein balance is lower. So we're using more protein in a reduced carbohydrate diet. And that makes sense because we see here we're using slightly more protein in the we're oxidizing slightly more protein in the reduced carbohydrate diet. So this table then shows what was significantly different between the reduced carbohydrate diet and the reduced fat diet. If we look at body weight, there's a significant difference between the reduced carbohydrate and reduced fat. So in the reduced carbohydrate diet, compared to the reduced fat, they lost more body weight. So at first glance, you might think, oh, this is better. They lost more body weight. If they lost more body weight, then they'll have a lower BMI, and that was also significantly different. But let's look at percent body fat. There's no difference in percent body fat. So if there's no difference in percent body fat, what did they lose in body weight? Okay, let's look at fat mass. There's no difference in fat mass between the two. So if they lost more body weight, but fat mass is the same, what did they lose? Well, body weight is made up of body fat, body muscle, body water, bone mineral content, and organs. So after 2 weeks, they probably didn't lose any bone mineral content. They probably didn't lose an organ. So that takes us down to they either lost body fat, body muscle, or body water. But our analysis of body fat says this wasn't different. So a reduced carbohydrate diet either reduces body muscle or body water. Okay. Well, that kind of makes sense because think about glycogen. Glycogen has to be hydrated, and glycogen holds water. If we're on a reduced carbohydrate diet, we're going to use up our glycogen. If we use up our glycogen, we're going to decrease total body water. We also can look at our respiratory quotient. So we see that that's different, and we see that the RQ is actually lower, and that makes sense because they're burning fat, but they're also consuming more fat because they're on the reduced carbohydrate. We also see that the fat oxidation is different. So in the reduced fat diet, we are going to be incurring a higher fat imbalance, Where in the low carbohydrate diet, we're incurring a carbohydrate imbalance. And if we also look at our urinary nitrogen, it's significantly different. And the participants were losing more nitrogen in their urine compared to the reduced fat. This is higher protein oxidation. So finally, if we look at cumulative fat imbalance, it's significantly different, and a reduced fat diet results in a higher cumulative fat imbalance. So even though the people on a reduced carb diet lost more body weight, they didn't lose more fat, and they weren't they were in less fat imbalance, burning less fat compared to the fat consumed. So this means that even though people on the reduced carbohydrate diet lost more weight, they didn't lose fat because they're consuming a lot more fat. They're just losing either body water or muscle. I forgot that I made these little things. And an important one to think about is it might be partially driven by this difference in protein oxidation between the two, even though protein is being held the same. So if we look back at our macronutrient balance, we see that gluconeogenesis is part of both carb balance and protein balance, but it's not related to fat balance. So if we decrease carb intake, we're going to increase gluconeogenesis. If we reduce fat intake, it doesn't change gluconeogenesis. There's no gluconeogenesis related to it. So Kevin Hall's group did a very similar experiment again. They didn't call it reduced carb and reduced fat. They called it, low carbohydrate and low fat. But the principle is the same. And in fact, this is the data that you used on your abstracts. And if you remember from your abstracts, we saw something similar. But we also saw that the low-fat diet tended to consume fewer calories compared to the low carbohydrate diet. But the low carbohydrate diet lost more body weight. Well, actually at the end it's not the difference, but they lost body weight faster. Where the low fat diet lost at the end the same amount. There's no difference here between this last data point. P is greater than 0.05. So the low fat diet also lost body weight, but it lost it slower. The low fat diet also lost more fat mass than the low carb diet. So remember though, there wasn't a difference in body weight at the end. So if body weight is the same, who has more muscle mass or more water weight? The low carbohydrate diet lost as much body weight but less fat, so they must have lost muscle or water, probably a combination. If we look at fat-free mass, so fat-free mass is water, muscle, and organs. And again, we can ignore organs. We're going to assume in 2 weeks no one lost any organ weight. So they lost either water weight or muscle. And the low carbohydrate diet lost more water or muscle. Compared to the low fat diet. So based on these two studies, you should be thinking to yourself, okay, a low carbohydrate diet, you might lose weight faster, and you might even lose more body weight, although that was different between studies. But what you're losing on a low carbohydrate diet is fat-free mass, not fat mass. That's usually not our goal. We want to lose fat mass. We don't want to lose body weight because we're losing muscle. So we can look back to our carbohydrate metabolism to make sense of this. This diagram is diagramming glycolysis and gluconeogenesis. Remember that largely glycolysis and gluconeogenesis are very similar pathways, at some points exactly the same, but operating in opposite directions. So glycolysis has 3 irreversible steps: glucokinase, phosphofructokinase, and pyruvate kinase. Gluconeogenesis has 4 irreversible steps: glucose 6-phosphatase, fructose bisphosphatase, and then what I'm just going to call PepCKC, which means cytosol, and PepCKM, which means mitochondria. And there's also this one we'll get to in the next slide, which is pyruvate carboxylase. Oh, I forgot that I wrote it right here. This is PepCK stands for cytosolic phosphophenylpyruvate carboxykinase. You can see why I only remember it as PepCK. And we also have fructose 1,6 bisphosphatase, which we'll get to in a moment. So this again comes back to insulin and glucagon. So for our pyruvate carboxylase, just to remind yourself, pyruvate carboxylase is here. It's part of gluconeogenesis. Its expression is increased during low glucose concentrations. So if we don't have enough glucose, that would mean that glucagon is high. So glucagon increases pyruvate carboxylase. So glucagon increases gluconeogenesis. And glucocorticoids increase pyruvate carboxylase. And we know that glucocorticoids increase gluconeogenesis. It's through this mechanism. Insulin will decrease the activity of pyruvate carboxylase because if we have high insulin means we want to store things. It means we have nutrients to store. If we have nutrients to store, we don't want to make more nutrients. And we don't want to break down stored nutrients. So we don't want to do glycogenolysis, the breakdown of glycogen, and we don't want to be doing a lot of lipolysis. And we don't want to do gluconeogenesis. All right, now I'm going to erase this because I know there's more coming. And also, pyruvate carboxylase is going to work to maintain TCA cycle intermediates. So when we don't have enough nutrients, we don't want to just burn up everything that's in the TCA cycle. We want to maintain those intermediates so that the cycle is ready to go right when we get more nutrients. All right, so now we have our PepCK cytosolic. Again, this is a gluconeogenic enzyme. Glucagon is going to increase it. Glucocorticoids are going to increase it. Insulin is going to decrease it. Insulin wants to decrease gluconeogenesis. Okay. Now let's look at fructose 1,6-bisphosphatase. Fructose 6,1,6-bisphosphatase is rate limiting. It's also sensitive to cold, which is more of an interest than something you really need to remember. But what's important is that fructose 1,6-bisphosphatase interacts with fructose 2,6-bisphosphatase. This is another enzyme. Insulin increases fructose 2,6-bisphosphatase. Glucagon decreases fructose 2,6-bisphosphatase. That's because this enzyme is part of glycolysis. It stimulates phosphofructokinase. So let's think about this for a minute. Fructose 2,6-bisphosphatase activates phosphofructokinase. Oops, sorry about that. And phosphofructokinase is in the glycolytic pathway, glycolysis. In fact, hexokinase is above it. It goes to phosphofructokinase. And then the next rate limiting step is pyruvate kinase. But anyway, so fructose 2,6-bisphosphatase activates PFK. And so insulin is going to increase it. Glucagon is going to decrease it. And it acts in opposition to fructose 1,6-bisphosphatase. All right, last but not least, we have G6 phosphatase. So phosphatases remove phosphate groups. Kinases add phosphate. When we have high concentrations of glucose 6-phosphate, it's going to increase glucose 6-phosphatase. High glucose 6-phosphate is increased by glucagon, glucocorticoids, and thyroxine. So this, again, this is promoting gluconeogenesis. It's removing a phosphate from glucose 6-phosphatase, which means it's making glucose that can leave the cell. That's going to increase blood glucose. So let's apply now what we've learned about these enzymes to the data we saw in Hall's experiments. He also took glucose measurements over time, and over the course of 360 minutes, this is an oral glucose tolerance test, we see that on the low-fat diet, so at 0, this is baseline, right about 10 minutes, they don't have a line for it, oof, it's a bad line, they drink a glucose drink, And on the low-fat diet, after drinking that glucose drink, their glucose increases. But on a low-carbohydrate diet, we don't see that increase in glucose concentration. That's kind of unusual. You would think a low-carbohydrate diet, you would think we'd see a dramatic increase in glucose. So this is our first indication that something odd is happening. Because after this, they largely normalize, although the low-carbohydrate diet is consistently has lower blood glucose. That makes sense because it's low carbohydrates. But these people came in fasted, and then they drank a glucose drink. Why don't we see this glucose spike? We need to look at more data. All right, so next, let's look at insulin. Okay, in the low-fat diet, again, this is the same glucose tolerance test, so at 10 minutes, they drank a glucose drink. We see another spike, and this time it's a spike in insulin. But we don't see a spike in insulin in the low-carbohydrate diet. We do in the low-fat diet. The insulin spikes, and then it goes back down. And there's no difference at the latter half. You might be thinking, okay, well, hyperinsulinemia is bad, so maybe it's good that insulin didn't spike. And, I mean, the glucose didn't increase, so it makes sense that insulin wouldn't spike because it's not sensing that excess glucose. And C-peptide, similarly, C-peptide is co-released with insulin, and we see, okay, C-peptide is released, but C-peptide isn't released on the low carbohydrate. And if we look at lactate, again, lactate increases in the low-fat but not low-carb diet. Lactate is a byproduct of glycolysis. So glycolysis is happening here, but it's not happening here. And that's odd. And if we look at free fatty acids, the free fatty acids decrease, but they only decrease a little bit in the low carbohydrate. Remember that insulin equals storage. We don't see an insulin spike in the low carbohydrate diet. We see less storage. We do see an insulin spike in the low-fat diet. We see increased storage. And last but not least, in the low carbohydrate diet, we see much higher serum triglycerides. But we don't see an increase in serum triglycerides in the low-fat diet. So if we're increasing serum triglycerides, that means that we are doing probably de novo lipogenesis, which means of the carbohydrates we have, even though it's low, we're turning them into fats, and we're consuming fats, and we're putting them into the bloodstream. Whereas in the low-fat diet, we're not doing that. So, I don't know why this won't move. If we go back, what this is indicating is that we need insulin. Oops. No, come on. I should have erased this initially. We need insulin because we need insulin to turn off gluconeogenesis. In a prolonged low carbohydrate diet, we have excess gluconeogenesis. That means we're breaking down fat-free mass. It also means we're breaking down our glycogen. And so body weight loss is really water loss and muscle loss. So the answer to which diet is better, if we're looking just to lose body weight and we don't care where that body weight is coming from, you're going to see faster body weight loss with a low carbohydrate diet. But if we want body fat loss, we need a low-fat diet. And ideally, a low-fat diet is going to spare muscle mass. If we spare muscle mass, what's going to happen to resting energy expenditure? It should be better. And if we have more muscle mass, this is going to make exercise easier. So that concludes carbohydrates and fats. Next lecture is we're going to dive deeper into protein metabolism.